Okay, so our lecture today is about converting amino acids to specialized products. So the objectives is the synthesis of thyroid hormones, melanin, and catecholamines from tyrosine, synthesis of serotonin from tryptophan, histamine from histidine, creatinine from arginine and glycine, and the therapeutic uses of L.DOPA, monoamine oxidase inhibitors, SSRIs, and antihistamine. Okay, so the amino acids that are important for the structures, they're highlighted. So tyrosine, and then glycine, tryptophan, arginine, and histidine. Okay, so basically, our human body, we have proteins. They're degraded and they are resynthesized continuously. So we always have a storage of 3 amino acids, and they're used for multiple formation. So they form metabolic energy, which is basically ATP. They form protein, and they form other products, which we're going to discuss today. So melanin, catecholamines, several coenzymes, purines, and pyrimidines. My laptop is lagging a little bit, so I'm sorry for not changing the slide. Okay, so thyroid hormones, they are derivatives of tyrosine. Basically, there's 2 thyroid hormones, T3 and T4. So as you can see here, so the structure of tyrosine, so you don't actually have to know this, but to better understand, these thyroid hormones, they are made up of 2 tyrosine and the addition of an iodine. So the most important thing, the thyroid hormones, they are derived from tyrosine. Okay. Okay. Melanin is also made from the amino acid tyrosine. So what is melanin? It's a pigment that protects our skin from harmful effects of the sunlight. So it's made up of the amino acid tyrosine in the melanocytes of the epidermis. So basically, tyrosine, it will be oxidized to L.DOPA, and then L.DOPA will be oxidized to dopaquinone. Oxidation, it means that we're adding an oxygen. Okay, so by the enzyme tyrosinase. And then these oxidative products, they will polymerize. Basically, they will all join together and form melanin. So any defect in melanin, it will cause albinism. And the common defect is in the enzyme tyrosinase. So what's important here, and you have to know that melanin, it's the pigment that's protecting our skin, and it's made up from the amino acid tyrosine. Oh, and the defect in melanin production that is causing albinism, that's very important. It usually comes up in the exam. It did for us. I'm sorry for the PowerPoint. I don't know why it's... Okay. What are catecholamines? So they are the neurotransmitters like dopamine, epinephrine that mediate a variety of central nervous system functions. So basically when you're in fight or flight response. Okay, so what could be these responses? It could be increasing your heart rate, dilation of the bronchioles, it could be digestive system, increasing your blood pressure, and converting glycogen to glucose. So, and we're going to talk about this point later on. Okay, so catecholamine synthesis. So phenylalanine will be hydroxylated to tyrosine, and then tyrosine will be hydroxylated to L.DOPA, and then L.DOPA will be decarboxylated to dopamine, and dopamine will be hydroxylated to norepinephrine, and norepinephrine will be methylated to epinephrine. So we'll talk about it more. Okay. So maybe the other way. Okay. So tyrosine will be hydroxylated to L.DOPA by tyrosine hydroxylase, and it requires BH4. So that's like very important. It usually comes up in this reaction, tyrosine hydroxylase, what does it require? BH4. And then L.DOPA will be decarboxylized to dopamine, requiring PLP. And then dopamine will be hydroxylated to norepinephrine, and norepinephrine will be methylated to epinephrine. And it requires a methyl donor, which is the SAM. So it's basically written here, like what I've said, tyrosine is hydroxylated to L.DOPA by tyrosine hydroxylase, and it requires BH4. And tyrosine hydroxylase is the rate limiting step. And then L.DOPA will be decarboxylated to dopamine, requiring PLP. And dopamine will be hydroxylated to norepinephrine by dopamine hydroxylase, and it requires vitamin C and copper. And then norepinephrine will be unmethylated to epinephrine, and it needs a methyl donor, which is the S-adenosyl methionine. Okay. So as we've said, dopamine, norepinephrine, and epinephrine, they are neurotransmitters, and they are synthesized in the brain, and in the CNS, they will function as neurotransmitters. So epinephrine is synthesized in the adrenal medulla from norepinephrine, and as we've said, in norepinephrine, epinephrine, they are involved in fight or flight during stress, and outside the CNS, so they are regulating your carbohydrate and lipid metabolism, basically. So from the previous, picture, where the glycogen is converted to glucose. Okay, so here. So why does that happen? Like, so during stress, why do you need to convert glycogen to glucose? So you need more energy because you're in danger, right? You want to run. So you need glucose, you need the energy. And that's why during fight or flight, the neurotransmitters, they are degrading glycogen and they are converting it to glucose. So it has a role in fight or flight and regulating your carbohydrate and lipid metabolism outside the CNS. Okay. It's showing you the same thing. In tyrosine, it will be hydroxylated, and this is the rate limiting step to L.DOPA by tyrosine hydroxylase, and then L.DOPA will be decarboxylated to dopamine, and dopamine will be hydroxylated to norepinephrine, and norepinephrine will be unmethylated by to epinephrine by phenylethylamine and methyl transferase. Okay. Parkinson's disease. So it's basically a brain disorder where you're having shaking, stiffness, difficulty with balance and coordination. You're having uncontrollable movements, and it's due to the loss of dopamine producing cells. So the dopamine producing cells, they are not producing enough dopamine. They're causing these symptoms, and the treatment would be to give them L.DOPA. Now, why don't we give them dopamine directly? It's because dopamine does not cross the blood-brain barrier. So we're basically doing nothing if we give them dopamine. So Parkinson's, it's a brain disorder the dopamine producing cells, they're not producing enough dopamine, and we're having these symptoms of shaking, stiffness, difficulty with balance, and coordination. Most important thing, Parkinson's is associated with insufficiency of dopamine, and that the treatment is L.DOPA. And dopamine, it does not cross the blood-brain barrier, and that's why we give them L.DOPA. Okay. Degradation of catecholamines. So epinephrine and norepinephrine, they are degraded by monoamine oxidase and catecholamine methyltransferase. So they're degraded by these two enzymes, and they are reversible, so they could be in either order. So this enzyme first or this enzyme first, it doesn't matter. They can be in either order. And the product will be the vinyl mandelic acid, VMA. While dopamine, it will be degraded by the same enzymes, but the product will be homovinylic acid. So the most important thing, epinephrine, norepinephrine, they're degraded by these two enzymes, monoamine oxidase and cateomethyltransferase. And the product is VMA. While dopamine, the product is homovinylic acid. And the products, they are an indication of how much norepinephrine is. And homovinylic acid, it is an indirect measurement of how much dopamine there is. So basically, this is what I've said, but written. So catecholamines, they are degraded by monoamine oxidase, by oxidative deamination, and by cateomethyltransferase, by O-methylation. And this enzyme, it needs a methyl donor, so SAM. And the reaction, as I've said, it occurs reversibly, so it can occur in either order. The products of epinephrine and norepinephrine, it's VMA. And the product of dopamine is homovinylic acid. Now, as I've said, VMA, it's a indication of how much norepinephrine and epinephrine there is. So in pheocytochromas, which is a tumor of the adrenal gland, basically there is more production, increased production of epinephrine and norepinephrine, which causes an increase in the VMA. As there is more synthesis of epinephrine and norepinephrine, there's more degradation, so there's more increase in the product, the VMA. Do you guys have any questions at this point? Was I going too fast, or does anyone have any questions? Okay, so SAM is a high energy compound, and it's important in metabolism because it can serve as a donor of which of the following metabolites? Yes, so it's C, yes. So it's as a methyl donor. Okay, which of the following is the coenzyme for phenylene hydroxylase? Okay, I think yes, THP. So if we go back to this one. So in the synthesis of catecholamines, phenylene will be hydroxylated to tyrosine by phenylene hydroxylase, and this reaction will require THP, which is also the same as BH4. So if they get you THP or BH4, they're the same things. Don't get confused. Okay. Serotonin, it's also a neurotransmitter, but it's not a catecholamine. So it has similar it functions as a neurotransmitter, and it's important in controlling appetite, your sleep, your mood, and cognitive ability. Now, it's synthesized from tryptophan. It will be hydroxylated to 5-hydroxytryptophan and decarboxylated to serotonin. The first reaction requires THB or BH4 tetrahydrobiopterin, and then the second reaction requires PLP. It also has the same, so it has the same pathway as dopamine. So tyrosine will be hydroxylated and then to L.DOPA, and then L.DOPA will be decarboxylated to dopamine. So it's the same pathway as serotonin. So tryptophan will be hydroxylated to 5-hydroxytryptophan, and then 5-hydroxytryptophan will be decarboxylated to serotonin. So that's basically what they mean here. You know, they undergo a similar pathway in requiring hydroxylation and then decarboxylation. Okay. So the largest amount of serotonin is found in the intestinal mucosa. It's a neurotransmitter in the CNS and in platelets. And as I've said, it has multiple physiological roles in appetite, sleep, temperature, cognitive functions, and mood. And this is why it's a good antidepressant. So to increase or to maintain the levels of serotonin, because it does affect mood, they use selective serotonin reuptake inhibitors as antidepressants. And in the pineal gland, it's converted to melatonin. Serotonin, because it's a neurotransmitter too, so it's degraded by the same enzyme, monoamine oxidase, as the catecholamines, right? Okay, so both dopamine and serotonin, they are degraded by monoamine oxidase. So monoamine oxidase, they are a strong class of antidepressants. Okay, so basically here, dopamine and serotonin, they both have, so serotonin, it's associated with feelings of happiness, focus, and calm. So it's associated with mood. And dopamine, it's also associated with feelings of rewards, motivation, and being productive. So both of them are good antidepressants. So monoamine oxidase inhibitors, they're basically inhibiting the degradation of dopamine and serotonin. So you have more, so you're preventing their breakdown. And they are more potent than SSRIs because SSRIs, they target serotonin, while monoamine oxidase, they are targeting both dopamine and serotonin. So they're more strong, they're more potent. Okay. In DHP R deficiency, so DHP dihydropyridine reductase, it's an essential enzyme for making tetrahydrobiopterin, so BH4 or THB, and it's needed for phenyl hydroxylase and tyrosine hydroxylase. So basically, if you don't have this cofactor, then these enzymes, they're going to have low activity. So basically, we're going to have less synthesis of catecholamines and serotonin because phenyl hydroxylase and tyrosine hydroxylase, they are in the pathway of the synthesis of catecholamines. So the treatment would be to have a low phenylene diet because you don't want phenylene to build up, because you don't have, basically you're inhibiting the action of phenylene hydroxylase. So phenylene would build up. So the treatment would be to have less phenylene and to provide the precursor for catecholamines and serotonin because you can't synthesize them. And so a deficiency in DHPR, you would have less synthesis of catecholamines and serotonin, and you would also have phenylketonuria, so basically a buildup of phenylene. Okay. Histamine, it's a chemical messenger that mediates a lot of cellular responses like allergic, inflammatory reactions. It increases gastric acid secretion and causes vasodilation. So how is it synthesized? It's synthesized from histidine by a decarboxylation reaction. And it requires PLP. Basically, when you have an allergic reaction or trauma, mast cells, they will release histamine. And so any agents that will interfere with the actions of histamine, they have important applications as antihistamine يعني medication مثلا. So you will stop the action of histamine, or you're downregulated if you take like antihistamine medication. Okay, so is everything clear till now? Okay. So as a physician with a background in the new field of metabolomics, you assess a 21-year-old with a classic phenylketonuria. The patient is fed phenylene with a label in the phenyl ring, and a 24-hour urine sample is collected. Which of the following compounds would you expect to collect the greatest amount of label in this urine sample? Yes, so phenylketone. So you're going to have a buildup of phenylene. And that's why it's going to be secreted as phenylketone. Yes. Okay. Okay. Phenylketonuria, alkaptonuria, albinism are caused by deficiencies in the enzymes involved in the metabolism of So basically, tyrosine, as we've said, the amino acid, it makes melanin. And so if there's a deficiency in tyrosinase, you will have albinism, alkaptonuria, and phenylketonuria, because it makes melanin, and then it's also involved in the buildup of phenylene. So it's tyrosine. Okay. Aspartame must be restricted in phenylketonuria patients because it contains BS. So basically, they have all the same concepts. They're pretty easy. So a small recap. So amino acids, they are involved in the generation of metabolic energy, ATP, protein synthesis, and specialized products that we're talking about. So we talked about tyrosine. Thyroid hormones, they are derived from tyrosine. Tyrosine also makes melanin. And a defect in melanin production will result in albinism. And the common type of defect is tyrosinase, so the enzyme. Then we've talked about catecholamines, which is also synthesized from tyrosine. And wait, we'll go to the okay. So tyrosine hydroxylase or BH and we talked about the function of the neurotransmitters. So epinephrine, it's synthesized in the adrenal medulla. The epinephrine and norepinephrine, they have a role in fight or flight reactions in the CNS, and outside the CNS, they have a role in carbohydrate and lipid metabolism. And then we discussed Parkinson's disease. So it's a brain disorder that has insufficient dopamine production, and the treatment would be L.DOPA. And then they are the catecholamines, they are degraded by two enzymes, monoamine oxidase and COMT. So the products of epinephrine and norepinephrine is VMA, and the product of dopamine is HVA. Increased VMA would be in pheochromocytomas, where there would be excess production of catecholamines. Okay, and then we discussed serotonin. So it's synthesized from tryptophan, and it has a similar pathway to dopamine. Okay, and then we discussed serotonin and dopamine. If we inhibit their breakdown, we can use them as antidepressants. So basically, they are strong class. Why? Because they're preventing the breakdown of both dopamine and serotonin. Being more potent than SSRIs, inhibiting more than one neurotransmitter. Okay, and then we talked enough about this. بعدين histamine is synthesized from histidine through decarboxylation. And histamine is released by mast cells when there's an allergic reaction or a trauma. Okay. Now to the last part. So creatinine. Okay. So creatine is synthesized in the liver and the kidneys from arginine and glycine, and you need a methyl donor. So arginine, creatine, and it will be reversibly phosphorylated to creatine phosphate through the enzyme creatine kinase, using ATP as the phosphate donor. So basically, arginine, glycine, and a methyl donor will synthesize creatine, and then when you add to it phosphate through ATP by creatine kinase, you will get creatine phosphate. And the presence of creatine kinase in the plasma will indicate heart damage and myocardial infarction. So this is very important. Yeah, it usually comes up, and it's a marker for heart damage and myocardial infarction. So basically, it's the same thing here. In arginine, glycine, and a methyl donor, they will make creatine. And then by creatine kinase, you will, by adding a phosphate through the enzyme creatine kinase, you get creatine phosphate. So phosphocreatine, it's the phosphorylated derivative of creatine found in the muscle. It's a high energy compound, and it's rapidly mobilized. So if you need energy and quickly, they can use creatine phosphate as a high energy compound to maintain the intracellular ATP levels. So as I said, during the first few minutes of intense muscular contraction, so when you need the energy quickly, they may use creatine phosphate as a reserve of high energy phosphate to maintain the ATP. And then the amount of creatine phosphate, so it's proportional to the muscle mass, okay. And then creatine and creatine phosphate, they will be spontaneously cyclized, so not enzymatically. They will just cyclize and form creatinine to be excreted in the urine. And the creatinine concentration, it's a marker for kidney function. So let me recap. Wait, from the picture. So arginine, glycine, and a methyl donor, you will get creatine, and then you will add to it phosphate through the enzyme creatine kinase, and you will get creatine phosphate. So where did you get the phosphate from? From the ATP. Now, how do you degrade it? Basically, it will cyclize to form creatinine, and it will be excreted in urine. So not enzymatically. And then phosphocreatine, so creatine phosphate, it's a mobilized reserve of high energy phosphate. So basically, if you need ATP really quickly, you use creatine phosphate. Okay, same. And the creatinine, it's a marker for kidney function because it's neither absorbed nor nor secreted. So you will have, so it's a good marker of kidney function. A newborn has milky white skin and white hair, a disorder that is caused by deficiency of which of the following enzymes? Tyrosinase, yes. Okay, next question. Which of the following is a metabolic product from the breakdown of muscle amino acid that is also used to measure kidney function? Yes. So that would be our last slide. Do you guys have any question? Tamam. Thank you.